Hi everyone, we are on to the data section of scientific papers, and as you probably know, data can be qualitative, which generally relates to sensory data that you pick up about the smell of a sample or the texture of a sample or the color of a sample, or quantitative data, which is measured numeric data. We use the metric system when we do those measurements. Researchers collect that qualitative or quantitative data in journals or in data collection tables. And the tables are in rows and columns for very easy reference and very systematic reference, especially when different researchers are doing the same experiment all over the world at the same time. They're collecting data into the same tables so that it is all uniform. When we look at analysis of that data, we generally will use several different ways to analyze it. We will analyze it using statistics, and those statistics can include sample size, standard error, p-values. You actually looked at percent error in the last lab. That analysis can fall into figures as well, particularly in biology, line graphs and bar graphs. And those line graphs are great for continuous quantities, continuous measurements. Continuous measurements are things like height and weight, which happen continuously. Bar graphs are better for categorical variables, categorical data like eye color or sex, where it's either one thing or the other. It's not on a continuous measure. Those are great for bar graphs. And then tables can also be used in your analysis to present your statistics that you calculated. So you can find that in your bar graphs as well. And just a moment, I'm trying to make sure that you don't get kind of crazy with, there, yeah. Now, great. I'm trying to make sure that I don't scroll because that can really get distracting. The major parts of those tables, as you know, from reading up on the file as well, are that tables in this CSE format have a table number at the top of the table and a title or a caption. This one is the absorbance of cobalt chloride on top of the table. The headings are the column titles, and they include the units with them. The body of the table is all the data. Sometimes a key may be necessary. The ones that you are seeing in your file do not have a key. And in fact, let me go down here a little bit. Yeah, that's an important point. In the tables along with the CSE format are the structure that the independent variable, the variable that you, the researcher, are controlling, is always on the left-hand side of the table. It's on this side of the table, the left-hand side. You are adjusting the wavelength in your spectrophotometer. And the dependent variable is always on the right side. Cobalt chloride will absorb that spectrum at a certain level, and that will be a dependent measure, a dependent variable. It will always be on the right side. These line graphs and bar graphs that we're going to talk about are visuals that present data very quickly, so you see it right off and you recognize the trends. They are not appropriate for tables which contain a lot more numbers. The figures are the visuals that help. They can be line graphs scatter plots, bar graphs, pie graphs, not so often in biology. Drawings, I use a lot of drawings. Gel photos, very specifically in biology. X-ray images, very specifically. Microscopic images, only when you're doing microscopy. So you will see a lot of line graphs and bar graphs in biological data. When it comes to a line graph, the key figures of a line graph are the figure number. I'm going to leave that up here a bit. You see the figure number here. And a description caption. This time, it's always below the line graph. The table had the caption above the table. The line graphs have the caption. This one is temperature in degrees Fahrenheit over a 7-day period in New Jersey. That is below the line graph. And the headings with appropriate units are on the axes. This is degrees Fahrenheit, the units, the body of the graph, and a key if necessary. In this case, we don't have a key. And I wanted to point out to you that the dependent variable is always on the y-axis, because the dependent variable is what is being measured, and it will have fluctuation that is caused by the change in your experimentation and your independent variable. So your dependent variable of degrees Fahrenheit here is always on your y-axis. The independent variable, which was controlled by you, you decided which days to measure the temperature. The dependent variable always goes on the y-axis for line graphs. Very important to remember that. The x-axis is for the independent variable. And the dependent variable is on the y-axis. The way I remember that for mnemonic, I remember do it yourself, DIY, right? So dependent on the y-axis, because you're doing these yourself anyway. If that helps you to remember it, I hope it will. And when we go to, we're going to be going to bar graphs and histograms next. Now remember, bar graphs and histograms are great for categorical data or when we're comparing categories to each other. They are really preferred in biology because not all your data will equal 100%. Pie graphs are for data that do equal 100%. You're looking at 100% of a pie and dividing it up. In biology, that doesn't always happen. Bar graphs really help us with that same categorical data that is different from 100% total. And those bar graphs can either line up with bars that are vertical or they can line up with bars that are horizontal. Let's talk about where those fit into each other. The decision on whether or not the bars are going to be vertical or horizontal really comes down to the description of the categories. If the description of the categories is very long, in other words, if this had said lemon juice at 2% concentration at 70 degrees, then you might be putting this on a horizontal bar, because you might be able to label that bar with that long title inside the bar. But generally, you're going to be labeling that right out here. And you're going to have more room to do that because you certainly don't have a lot of room down here to put all of this information in. So you can, when you have an extremely long description, you can move the bar over to the right, and you can take more time on a horizontal, you can take more space on a horizontal bar to give yourself more space to write out the description. Otherwise, generally you're going to see vertical bars on bar graphs. Same thing on bar graphs. you're going to be having the figure underneath the bar graph, and you're going to have the title underneath the bar graph, and you are going to have the x-axis as your independent variable, and you're going to have your dependent variable on the y-axis. You are also going to have your axes labeled, and you will always need units. Now, placing the bars is an art in itself. Normally, you place the bars sequential to the procedure, which means that in this case, you tested lemon juice first, you tested baking soda second. And normally you work in the procedure's time frame and how first thing, second thing, third thing, fourth thing. If you are recording everything at more or less the same time and it doesn't fit into a time frame in the procedure, that can happen. You generally go from the shortest bar to the tallest bar. Or if it makes sense, you can go from the tallest bar to the shortest bar. You want some kind of a sequence to this, either a procedural sequence, shortest to tallest, or tallest to shortest. You don't want it all over the place unless that is part of your procedure that is very well spelled out in a bar chart. When you do a bar chart, these bars all stand separately. They are not smushed together, and they are separate, and the bars are narrower than the space between them. So it's very easy to read the bars because there is so much space between them. So these are narrow bars, lots of space between them. And I guess I already did that. I already talked about that too. As for the conclusion is the final part of the scientific method. It evaluates the hypothesis. It decides whether or not your hypothesis is supported by data that you collected or refuted by data that you collected and analyzed. And if the data supports the hypothesis, and you hope it does, then you can discuss in the hypothesis testing additional variables next time, or your conclusions on what you would like to test next time, how you would like to proceed with this, or you can start writing your paper for publication. When you are thinking about a conclusion, you do not simply regurgitate all of your findings. Your findings should be spelled out with your analysis. So your conclusion is really supposed to highlight your hypothesis and whether or not the data supported or refuted the hypothesis. If the data does not support the hypothesis, or if the data is unclear whether or not it supports the hypothesis, you can insert in your conclusion some information about additional trials that you would do, what you would change about those trials, any revised procedures, any new hypotheses, any new research that you think you would need to do before you started on another experiment. Scientists do not worry about failing. There's no such thing really as failing in science, because you go back and you learn and then you employ that in your next trial. So scientists are very open about discussing their data that does not refute that does not support the hypothesis, and in fact might refute the hypothesis, because all data is important and can lead us toward more educated predictions for hypotheses in the future. See you on the next slide.